Prathamesh, is it on?
Yes, sir.
Is it on?
Yeah, it is being live streamed.
Please let me know.
Hello.
Hello. On हो गया?
Yes, sir. Hello.
Okay. Hello. Today we have a very accomplished speaker, Dr. Ruchira Sen, who will give us an excellent talk. She did her studies from IISc, Bangalore, which is a premium institute in India. Now I call upon Dr. Sonia Bhatta, who is a life member and the coordinator in Chandigarh. Sonia. Dr. Sonia.
Good evening to all of you. First of all, I want to thank Dr. Swaranjit Singh for providing me this opportunity to introduce himself and our speaker, Dr. Ruchira Sen. So, little about Sir. Sir name is in the list. Sir's name is listed in the top 2% global scientists for 3 consecutive years. And Dr. Swaranjit has served in MTCC and Gene Bank, now the first IDA in India. He is member in 2 WFCC committees. He is member Task Force National Biodiversity Authority, Chennai. He has been awarded as Loyola Environmental Award, Lifetime Achievement Award 2022 by St. Xavier College, Ranchi, Lifetime Achievement Award 2023 by Shulini University this year in 2023. His publications, they are in high impact factor journals, and the articles, they are highly cited in the literature with H index 56, I20 index 150, and he has more than 17,000 citations, and his name is in the top 2% global scientist. So we are so lucky to have you as our mentor, and we will work under your guidance, sir. Thank you so much. Now I will like to introduce my colleague, Dr. Ruchira Sen. And she is an assistant professor in the postgraduate department of postgraduate department of geology at Shri Guru Gobind Singh College. She had done her PhD from Indian Institute of Science, Bangalore. She then did 2 postdocs from Universities of Texas at Austin and Purdue University at West Lafayette, Indiana. She has worked as on social insects like wasps, ants, and termites. Her major research projects are on behavioral ecology, microbial ecology, and molecular ecology. She has published more than 24 research papers in reputed international and national journals, and she is funded by grants from SERC more than 39 lakhs. Today, she will talk about symbiotic mediated crop protection in social insects. Over to Dr. Ruchira. Thank you.
Thank you so much, Sonia ma'am. Am I audible?
Yes, you are.
Yes, ma'am.
Okay, so today I'm going to talk about the dynamics of symbiont-mediated crop protection in social insects. I am really grateful to Dr. Swaranjit Singh for giving me this opportunity to share my work with you all. And it feels really nice to join the Microbiologist Society in Chandigarh. And we are happy to have this platform to work on microbes as well as to share whatever work we have done with all other peers. So without further ado, let me start. The dynamics of symbiont-mediated crop protection in social insects. Now, when we talk about crop, we actually associate this word with agriculture, because agriculture is production of food and in a systematic manner. A large-scale production of food, it can be crops, it can be domesticated animals, etc. But you produce food sitting in one place or staying in one place. You don't have to move around, right? Now, we humans invented agriculture about 12,000 years ago. And this invention helped us from shifting from a nomadic hunting-gathering lifestyle to a settled civilization-based lifestyle. So agriculture had a very impactful role in our civilization. Now, agriculture, although we invented it about 12,000 years ago, there are some insect societies where agriculture has been invented much earlier than ours. Okay. Now, who are these insects who have invented agriculture? There are many social insects like ants, bees, wasps, and some beetles as well. But agriculture has been invented by certain ants and some termites. And this agriculture has been invented about 50 to 60 million years ago. So you can see that although in our civilization or our human history, agriculture played a very important role, it is a very young invention. Whereas in social insects, those who have evolved it, in their lifestyle, in their societies, agriculture has been a part of it for a very, very long period. Now, you might have seen this picture in National Geographic Channel or even in movies like The Lion King, where a trail of ants are carrying some small pieces of leaves and like pieces of flags on their head, right? So this is a very common picture or a common video in many natural history channels. What is this? Why are these ants cutting leaves? These ants do not feed on these leaves. They cut these leaves from big trees and then they take these pieces of leaves to their home or to their nest and they chew it or mash it up to make it into a fungus bed where they seed some fungus granules and they farm that fungus as food. So these insects, they do not, although they cut leaves all the time, they do not eat anything else other than the fungus that they grow. So you can see that it is very similar to agriculture, right? What we do. Now, there are some beetles who also show this fungus farming as their mode of food production. But we are not going to talk about the beetles today because beetles are in true sense not considered as social insects, or they are not as social as ants and termites. So we are going to restrict our talk to ants and termites. Now, where are these amazing fungus farming insects found? It has been shown that the fungus growing ants are found in the southern part of North America, in South America, and that is it. It is restricted to the New World. Whereas fungus growing termites are found in Africa and Southeast Asia. Australia is kind of safe from all these pests, but and why I say pests? Because although they are growing their own food, to grow their own food, they destroy our property. So if termites are there in agricultural fields, they spoil our agricultural fields, then they spoil the roots of wheat or other such crops. And the fungus growing ants, especially the leaf cutter ants who also form fungus, they can actually spoil the trees. Like they can cut off the leaves in no time. Like they can clear out all the leaves from very big trees. So they are many in number, and when they attack together, they can just shave off the trees in very little time, right? So they are considered as major pests in both these parts of the world. Now, let's compare our agriculture with the insects agriculture. There are many steps of growing crops, like acquiring the seeds and choosing the right kind of seed, then preparing the soil, sowing the seed to the soil, then adding manure and fertilizers, removal of weeds and pests, irrigation, harvesting, etc. Now, each of these steps are very important, but today I'm going to talk about the removal of weeds and pests, because even if you do all the other steps properly, just if you ignore this removal of weeds and pests, then your agriculture can be totally wasted, right? Your crop will be wasted. You won't be getting any food from your crop. Okay. Now, let's come back and we can show that each of these steps are also very important in insects agriculture. Okay. Now, I will only, like for the time limit, I'm only going to talk about the removal of weeds and pests in social insects agriculture. But before I do that, let me talk about what are these insect farmers getting from this agriculture? Why do they practice this? Okay, so insect farmers basically give nutrition, shelter, disease protection, etc. to the fungus, and the fungus gives food to the insect farmers. So in our agriculture, we cultivate different kinds of crops like cereals, pulses, vegetable, fruits, etc. But all insect farmers, they restrict their agriculture to fungus cultivation. Okay, now this is a picture of an Atta texana leafcutter ant in its fungus bed. You can see that this huge insect is their queen, and all the smaller ones are the workers. So the queen's main function is to lay eggs, and the workers make the garden. They sow the seeds of the fungus and they cultivate the fungus. They continuously grow their garden by adding smaller pieces of leaves to it, and they maintain the fungus properly. Now, here you can see that there is a symbiont association. So the insects have a symbiotic association with the fungus. So they are giving shelter and food and protection to the fungus, and the fungus in return is giving food to the insect. But this is not the only symbiont association in this system. There is also a symbiont association with a set of bacteria. So why do they need this symbiont association with the bacteria? For that, we need to talk a little bit more about the fungus and the ants association. So we have already said what the fungus is getting, what the ants are getting. But in any successful symbiotic association, there are often many parasites who exploit this symbiotic association. And true enough that even in attine ants colonies, if we isolate fungus from there, we see that although the ants are putting all their effort to cultivate a particular type of fungus, which is known as the Atomyces fungus, there are traces of many other fungi like Trichoderma, Fusarium, Syncephalastrum, etc. But they all remain in small quantities. But most important of all of them, or the most dangerous of them, is one called Escovopsis. This Escovopsis is one of the fastest growing pathogens. And when the ants are there on the colony, they keep the Escovopsis under check. But if you remove the ants from a colony, then you would see that Escovopsis would grow very fast and will take over the whole colony in no time. Okay, so for this Escovopsis control, the ants have certain methods. Now think about our agriculture. When we need to control the pests, when we need to control the weeds, what we do is we use chemical pesticides, we use biological control agents, we use IPM, and so on. Now, what do the ants do? The ants have something very similar to biological control agents. In a groundbreaking paper in 1999, Cameron Curie et al. Showed that these fungus-growing ants use antibiotic-producing bacteria to control garden parasites. What they showed is that the fungus-growing ants have a culture of pseudocardia bacteria, which is a type of actinomycete bacteria. They have a pseudocardia bacterial culture under their prothorax. So this part of the insect's body is known as thorax, and below the prothorax, or the first segment of the thorax, they have a culture of pseudocardia bacteria. So this pseudocardia bacteria, they showed that it can be used to protect the colony from Escovopsis. Because what they showed was that if you use this bacterial secretion, then Escovopsis cannot grow. So their paper was based on this ant Escovopsis pseudocardia co-evolution model. And this model was based on 4 key observations. Okay, so they made a model where they showed that ants are using pseudocardia bacteria to control Escovopsis fungus, which can destroy their cultivar fungus. Right? So their 4 key observations were: number 1, there was a single lineage of pseudocardia in an ant colony. So workers from one nest were associated with only one pseudocardia lineage. So they showed that only one pseudocardia strain was specific to the colony, and there was vertical transmission. What is a vertical transmission? When new queens are made in a colony, then they will have the same pseudocardia strain with them. And when they start their new colony, they will carry that pseudocardia with them, and that will protect the new colony. So mother to daughter, daughter to granddaughter, this way there is a vertical transmission of the same pseudocardia strain. Now, if the ants were getting the pseudocardia strain from somewhere else, that would be a horizontal transmission. But here they showed that there is no chance of horizontal transmission. Then they also showed that this bacterial secretion was actually promoting the growth of cultivars. So cultivar is the Atomyces fungus, and the secretions from the pseudocardia bacteria was promoting the growth, and it was inhibiting Escovopsis growth. So you see how beautiful this model is, and if it worked, then it would really work well to protect the agriculture of the ants from the Escovopsis. So it is specific to the colony. It is being vertically transmitted from mother to daughters. It promotes the cultivar growth but inhibits the pathogens growth, right? So I'm going to divide this key observations into 2 parts. And we did a study where we showed that this model was not as good as it sounded. So to do that, to verify their results or to do it at a larger scale, what we did was first we isolated and characterized the actinomycete bacteria from different nests of fungus growing ants through culture dependent and culture independent methods. So for the culture dependent methods, we used these 6 different species of ants, and they're like 1 or 2 colonies, sometimes 5 colonies. And these are the colonies. Some of them were collected from the field, and some of them we already had in the lab growing for a very long time. So what we did was we collected these colonies. From these colonies, we collected the bacteria and we cultured them on plates, and we saw that what is the identity of the different types of actinomycete bacteria. Because if that model was true, then every nest should have a different strain of Pseudocardia bacteria, right? So to do that, what we did was we collected the ants and put them in sterile saline buffer and then vortexed it. So when we vortex it, it is basically moving the Eppendorf tube very fast, and all the bacteria that is present on the integument of the ant or on the cuticle of the ant, that would be dislodged into this saline solution. And then we took that saline solution and we spread it on a low nutrient media. So this is known as the chitin media plate, and actinomycete bacteria, if you know about them, they can grow on very nutrient poor places. Okay, so we used a nutrient poor media so that we could only get the actinomycete bacteria, and all the other bacteria that needs a nutrient rich medium would be inhibited from growing in these plates. And actinomycete bacteria, they grow very slowly. So we wrapped these plates and kept them at room temperature for about 2 to 3 weeks. And in 2 to 3 weeks, we had small colonies of different bacteria growing on these plates. So we collected each of these colonies and then we stripped them on separate plates. What we showed is that there are many different strains of Pseudocardia. So we call them actinomycete morphotypes, and there are different types of strains here. These are all Pseudocardia strains, and these two were Amycolatopsis strains. So Amycolatopsis is also an actinomycete bacteria, but it was not reported earlier from these colonies, and we showed it for the first time. Now, if you look at the Pseudocardia lineage, if you look at the phylogenetic tree of Pseudocardia and other Amycolatopsis bacteria, you see that they can be divided into groups like Alni group, Compacta group, Halophobica group, Thermophilia group, etc., based on the main representative of that group. So there are, you can see that there are different clades of bacterial groups, and we show that these different strains that we found were representative of different groups. Okay, now this table is showing you here the colonies from where the bacteria were isolated, and these are the different morphotypes that we found. And here we have written W, G, M, F, etc. W means worker. So this Pseudocardia from Compacta group, this strain was isolated from Mycocyperus smithii ants and workers of different colonies. This is how we read this table. And this particular Pseudocardia strain was found from worker males, females of Siphomirmex wheeleri, then again from Mycocyperus smithii males, smithii colonies. So you can see here that although the first paper showed that every colony has a specific strain, we are finding the same strain from different colonies of different species. So this strains being specific to a particular colony was not true. Okay, then we isolated and characterized Pseudocardia from different nests using culture independent methods. So what we did was we used 454 pyrosequencing method, and this was a method very popular at that time. And we used this system, and we were the first ones to study insect microbiota. So what this system does is that it sequences all the bacterial strains at one shot. Okay, I'm not going to go into much detail about what 454 pyrosequencing is, and it has now become obsolete. This study was done in 2009. Now we use nanopore platform to do similar kinds of assays. But at that time, 454 pyrosequencing was a very in thing, and we used that to do a culture independent study. And for that, we took Mycocyperus smithii ants, Trachymyrmex septentrionalis ants, and Siphomirmex wheeleri ants. So these are the representative colonies. And one thing I would like to mention here is that Trachymyrmex septentrionalis nests are not prone to Escovopsis attack. Okay, but the other two colonies are. So we found quite a lot of operational taxonomic units, and this is the richness of that from different colonies. And these are the rarefaction curves, which basically means that we had sequenced it long enough, and we had like each of these curves are plateauing. So this basically means that we had sequenced enough, and we have found almost all the operational taxonomic units that were there. Operational taxonomic units here I'm saying as a representative of different strains. Okay, now this is called a heat map. In the heat map, what we are showing here is that at the bottom you can see that these are different nests that we used for this culture independent method of identification. And these are the different strains of bacteria that were more prominent in these nests. And blue means that these particular strains were not present in a sample, and red means that they were present at very high amount. Now, you can see that pseudocardia here, there are two strains which are present in, like multiple strains are of pseudocardia were present in the same colonies. You can see here as well that there were 3, 4, or basically more than one strains were present in each of these colonies. Then we we made a heat map with only Pseudocardia and other Amycolatopsis strains which produce antibiotics, and we showed that there are not only Pseudocardia strains, but there are quite a few Amycolatopsis strains as well, which were present in addition to Pseudocardia. So if you are saying that, okay, Pseudocardia is the one which is controlling the Escovopsis, we are showing that there are other bacteria which has the potential to secrete similar antibiotic secretions. Now, with culture independent 454 pyrosequencing data, we confirmed the results that we found through our culture dependent studies. So in this model, we showed that workers from one nest were, we kind of revised the results. So workers from one nest were associated with only Pseudocardia lineage. This was not true. There were multiple lineages. Vertical transmission was not the only mode of transfer, because males also had Pseudocardia. So when the male mated with the queen, there was a chance of horizontal transmission as well, because the male and the female were coming in close contact, and these bacteria grow on the cuticle, right? So the bacteria could very well come from the males of a different colony. Then it promotes cultivar growth and it inhibits Escovopsis growth. That part I am going to show you now, that whether it holds true or not. So we did some antibiotic challenges with Pseudocardiaceous secretions and different fungi. So we took these plates, these are nutrient-rich plates, and here in the center, we strict a specific strain of Pseudocardia or Amycolatopsis, and then placed 2 monocultures of Atomyces, fungus, or Escovopsis, or other entomopathogenic fungi, or other fungi that were found in the colony. They are monocultured in 2 sides of the bacterial streak. So this is the bacteria. And here we placed different types of fungi which were isolated from the ant colonies, or they were collected from other institutes which were known entomopathogenic fungi, which basically means they attack insects, right? So we saw that there were different kinds of growth of these fungi in these plates. So there were some fungi which did not grow at all, okay? So we would call them, there was no growth. Then there were some where the fungi grew a little bit, and we call them attenuated growth or arrested growth. Then sometimes the fungus would grow till the bacterial culture, but then it would stop. So we would call it the bacteria touch growth. And then sometimes the fungus would grow completely all over the plate without caring where the bacteria is. So we would call it as the full growth. So if you look at the effect of the antibiotics, then this one, that is where the fungus did not grow, we would consider it as the bacterial secretion had such inhibiting effect on the fungi that the fungi could not grow at all. And here, there was no effect of the antibiotics on the fungus, and fungus could grow even over the bacterial growth. Sometimes the attenuated growth looked like this, that there was a clear zone of inhibition at the center. So this set of pie charts, they are showing different kinds of inhibition profiles. Okay, so sometimes if it is completely red, then it means that the fungus could not grow at all. The pink means that they showed some kind of attenuated growth. Light green meant that the bacteria would touch the fungus, but after that the fungus would not be able to grow. And the dark green one shows the number of fungi which would grow even over the bacteria. Okay, so these are for this assay we had used 7 cultivar fungus to see whether the bacteria is actually promoting the cultivar growth or stopping the cultivar growth. We used 9 entomopathogenic fungi, 4 garden pathogens, and 2 endophytes. And you can see that there were very few fungi which had no effect by the bacteria. Most of them were inhibited by either the pseudocardia or Amycolatopsis. This one you can see is an Amycolatopsis. This one is also by Amycolatopsis. So not only pseudocardia, Amycolatopsis, which is another actinomycete bacteria, this one also had a strong effect on the fungi, and not just Ascovopsis, but it had a generalized effect on many different fungi. Okay, so we have to conclude the study. Workers from the same nest may carry several strains of pseudocardia bacteria. Culture independent survey strengthens our result from culture dependent survey. And we show that, yes, there are different strains of pseudocardia, Amycolatopsis, and other actinomycete bacterial strains. Males carry pseudocardia, and pseudocardia is not the only antibiotic secreting bacteria which is present on Atynians. So Amycolatopsis, which is a closely related antibiotic secreting bacteria, that one was also present in large quantities in many of these colonies. We published this work in 2009, and the first, obviously, we showed that the Nature paper, which had shifted how we thought about insect agriculture, was not completely true. And second reason why both these papers were important is that these papers started the studies on insect microbiota. Nowadays, many people are studying insect microbiota, but these were the pioneer papers on that field. So we also had another paper where we studied the microbiome of Trachymyrmex septentrionalis, where I already told you that it does not get Ascovopsis attack. But there also we found many different Amycolatopsis and pseudocardia, which basically says that like stopping or inhibiting Ascovopsis is not the only reason for having these bacteria. They might have some other effects as well. Now, here when we came back to India, we worked on a very similar study on Odontotermis obesus, which is a fungus growing termite. So in India, we do not get fungus growing ants. We get Odontotermis obesus, which has a parallel kind of agriculture. So they have Termitomyces fungus. They have, I mean, this is how like Odontotermis obesus mounds look like. It is very common in Chandigarh, Mohali area. And If we go to the colony, then we see that there are these termites. I don't know why this video is not working. Yeah. So you can see that their colonies have a little different type of a fungus garden. There, the fungus growing ants collect vegetative material and leaf to make the fungus bed. Here, the termites eat the vegetative material and use the semi-digested feces to make the fungus bed. And there, they cultivate termitomyces fungi. You can see these white small nodules. These are the fungi which are cultivated for food in termitomyces, in termites. Now, termites have exactly similar parallel thing going on here. And like Escovopsis, we have a parasite here or weed here, which is known as pseudoxylaria. So pseudoxylaria is also, we showed this in 2022, that pseudoxylaria was stopped by a bacteria called Pseudomonas. So pseudoxylaria is similar in nature like Escovopsis, but phylogenetically it is very different. Pseudomonas, which is another antibiotic secreting bacteria, it is having a similar effect on pseudoxylaria, but it is nothing like actinomycete bacteria. Okay, so they are very phylogenetically different, just like ants and termites are very different. Evolution wise, phylogenetically, they're very different organisms. Their fungus is similar, but the bacteria that they are using is different, but it is a parallel system. Okay, so this result was published in Microbial Ecology. Okay, now I learned all this work from Professor Ulrich Müller, who is one of the most famous persons who work on fungus growing ants. And this is my group who worked with me, or I worked with them on fungus growing ants. And this is the termite group in India with whom I collaborate. And for photos, I would like to thank Alex Wild and pixabay.com. And I would like to thank you for your patience and thank you for listening to me. If you have any questions, I would be happy to answer.
That was an excellent talk, Dr. Ruchira.
Thank you, sir.
I really enjoyed every bit of it because you talked about microbes and fungi. Because I love the microbial world very much. Now, if anybody has questions, they can ask. Any questions? Okay, if not, then I will ask a question. Dr. Ruchira, you said you isolated bacteria, especially actinomycetes, that produce antibiotics. What were the main strains you encountered?
Main strains?
Yes, main bacteria.
Well, Pseudocardia and actinomycete are the two. We found multiple strains of Pseudocardia as well as actinomycete. If you are interested to know about their sequences, all the sequences are submitted to NCBI, as well as the sequences were available in the lab. So if you need the strains, exact strains, those strains can be provided to you as well. But we gave them specific names. It was not possible to identify each of them, like what species is that, but we gave them numbers, and sometimes it matched with a sequence with NCBI, so we gave them that name.
All right, very nice. And the pseudonocardia looks very interesting.
Yes, it is. It is actually found in many different places. It is found in caves. It is found in on rocks, like moist places, or sometimes like dry, very hot weathers or climates. It is found in many places. But these ants, their cuticle turns out to be a nutritional poor medium which also harbors pseudocardia.
Okay. What temperature can this pseudonocardia tolerate?
Actually, actinomycete bacteria can tolerate very high temperature, but ants, as you know, that they stay in a very controlled environment.
Yes.
They live under the soil, so they are
Yes.
subterranean. So their colonies maintain a certain temperature, and when we kept them in lab, they were kept under air conditioning, so they were kept at around 23 degrees centigrade, but they can survive in high temperatures.
This work you did in or in USA?
I did at University of Texas at Austin.
These bacteria are also found in India, in Bangalore, for example.
It is basically found everywhere.
Okay.
But
Okay.
the ants do not have it probably,
Yeah.
but or else this kind of work has not been done much here in India. Nowadays, microbiota analysis is becoming a very hot topic. Many people do
Yes.
that, and it is much easier to do it. And nanopore platform is comparatively cheaper than 454 Paris sequencing. The data is easier to analyze. So nowadays, for termite work, we use nanopore platform, and we have some more papers coming up. We have worked more on this field here, but we haven't worked on ants. We have worked on wasps for this, but we
Yes.
haven't worked on ants. And you do not get fungus growing ants here. So since it is associated with agriculture, and our attention is towards seeing that how these symbionts can protect the crops of fungus growing insects, I don't see that pseudocardia would be available in many different insects, but
Yes.
if they do, I would not be surprised.
Right. So this is really very interesting and very amazing work.
Thank you so much.
We are short of antibiotics nowadays. You know that.
Exactly. So we can get more antibiotic secreting microbes from the
Yes.
social insects. Actually,
Right.
I'm thinking of, like, I'm in the process of writing a grant for that how we can isolate different microbes, which will be helpful in getting antibiotics, which will help us to control the more resistant
Yes.
antibiotics.
That's what I thought today on listening to your talk, that we are very short of antibiotics that are effective.
Yeah,
Most
exactly.
of them are drug resistant.
Yes.
Right.
Yes.
So this may be a very good source
Yes.
of new antibiotics.
Yeah.
So
Right.
we can discuss later on. I will discuss with you how this can be propagated.
Yeah.
That we should look for new antibiotics that are effective from insects.
Right.
This will be a great project.
See, social insects live in a group. When insects live in a group and in very close proximity, at that time, they are more prone to diseases. So
Right.
if they're more prone to diseases, they would need protection from the disease as well. So
Right.
you would expect more antibiotic secreting microbes in social insects.
Yes.
Right. So that is why, in fact, when I came back to India, that was my main project, like like for all kinds of job applications, I used that idea. But unfortunately, I didn't get a research-based job. So I'm teaching now mainly. So I do science using wasps now, or I collaborate with others. So that idea kind of has taken a backseat. But yes, it would be a great idea.
But really, your talk was very, 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 very good. Enjoyed it. Thank you so much.
Thank you.
So now I call upon Sonia Bhatta to please give a vote of thanks.
Yes, sir. Thank you, Dr. Ruchira Sen, for telling us about the symbiotic relationship between the bacteria and the fungi. And thank you, Dr. Swaranjit Singh, for providing us this platform to share knowledge about the different fields. Sir, I want to thank you about making one umbrella, bringing all the researchers from different streams under one umbrella. Thank you so much.
Sonia, thank you very much for the nice welcome and for the nice vote of thanks.
Thanks.
And I'm so proud to say that both of you Ruchira and yourself, Ruchira Sen and yourself are life members of MBSI. And
Thank
keep
you.
on your good work. Spread the subject.
Thank you.
Thank you so much.
Thank you
Thank
so much.